And to fully understand the strength and the durability of the immune response, you use a combination of molecular and immunological assays. So things like flow cytometry, what you're doing here is you're really characterizing your CD4 cells and your CD8 cells, because you want to make sure that these are being activated, which will ultimately lead to B cell formation. And so that's just a measure of activation that your immune system is doing what it's supposed to do, and you're getting the generation of markers, and then also the generation of B cells and memory formation. Then you have ELISpot, which allows you to detect T cell cytokines, in particular interferon gamma and IL-2 are usually the common ones that are used, and it really provides insights into the magnitude and specificity of the response. Again, multiplexing technologies are also used to measure biomarkers, which really to give you a comprehensive profile, immune profile, and really have a deeper understanding of some of the responses that are happening. And the traditional Western blots, detects proteins associated with T cell activation and confirming the presence of antigens produced by the mRNA. And then PCR, your traditional PCR to validate gene expressions linked to immune activation. Another one, as I said earlier on, single-cell RNA-seq. This is a technology that is rapidly evolving and will help look at the biomarker profile at a genomic level for using a single RNA-seq. And so by characterizing the immune response, whether it's humoral or T cell, they help refine the responses you get, will help refine and the vaccine formulation and optimize these dosing strategies. And this is why you need to have this information as you're doing development of your mRNA vaccines to ensure that there are no adverse effects. Now, like I said before, that's a lot of in vitro tools. You also got in vivo tools. You want to assess safety and tolerability of these mRNA-based therapies. And what in vivo allows you to do is to help identify some of these potential adverse effects, biodistribution and physiological impacts, to ensure that the mRNA and its delivery components are well tolerated. Because you don't want to be injected with something and you realize you're suddenly reacting to it, and you'll know, put it that way. And so one of the primary concerns with mRNA vaccines is really biodistribution. You want to ensure that its mRNA and its components, like lipid nanoparticles, reach the intended tissues, such as muscle cells or immune cells, without accumulating in non-target tissues to reduce toxicity risk. And by studying biodistribution, you can assess how long the mRNA and its components persist in the body and how they are cleared. And that's really about identifying any potential long-term safety concerns. And it'll help mitigate risks of off-target effects and adverse reactions, so that the vaccine and its components don't inadvertently affect other parts of the body. And to assess this, one of the common methods that is used is really imaging techniques such as luciferase assays, mRNA-loaded assays, in models such as mice, can also use non-human primates. And you're literally just tracking where the mRNA is distributing throughout the body. It's very similar to a PK assay in therapeutics, only in this case you're using an imaging technique as opposed to taking blood samples. And another method is commonly used is immunohistochemistry as well, where you can track mRNA movements, get down to a granular level, look at infiltration of cell types. Are there any adverse reactions? But beyond biodistribution, it's also important to assess the overall well-being of test subjects following mRNA administration. Key factors, and these are more physical, looking at behavioral changes. Are they behaving differently? The activity levels, are their feeding and drinking habits different? Their social, physiological, body weight, temperature, respiratory. These are probably going to sound very familiar when you're injected it as a human. your temperature goes up, but does it sustain or is it doing something else? It should only be temporary. And general health conditions, in particularly when you're doing preclinical things like coat conditions and eye and mucous membrane appearance and so forth. And so the goal of in vivo safety is really to identify and mitigate risks to ensure that it's the mRNA vaccine is being well tolerated and it has a good safety profile before moving on to clinical trials. Now, I think we've all heard this and we all knew about it. It was definitely something that was associated with COVID-19. One of the biggest concerns in infectious diseases and vaccine therapy is the potential of cytokine storms. It's a hyperactive immune response where the body releases an excessive, uncontrolled surge of pro-inflammatory cytokines, which can lead to severe inflammation and tissue damage, and in extreme cases, organ failure. And it has been observed in infectious diseases such as influenza, COVID-19, autoimmune diseases, graft versus host diseases, and certain medical conditions like CAR-T cell therapy. And the vaccines are really kind of designed to, in general, mRNA vaccines are really designed to trigger a controlled response, teaching the body to recognize and fight infections, and hence why you get that mild effect when you get vaccinated. And while vaccines stimulate cytokine release, this is kind of the normal part of the immune activation. And so the results, as I said, are typically mild, short-term, like fever, fatigue, and soreness at the injection sites. And this is what the body is supposed to do. It's mounting an immune response. Now, in theory, the risk of vaccines inducing those cytokine storms are very, very rare, thankfully, but could be of concern if there is an individual with a pre-existing inflammatory condition and it's not identified earlier on. So you have to be fairly rigorous in safety monitoring to detect any potential severe reactions that could be occur, and any potential, whether it's coming from formulation, which is often the case, as opposed to the mRNA, or if there's any pre-existing conditions. And this is just an example of another case study where non-human primate models are used, and they are highly relevant models for evaluating human vaccines due to their predictive immunogenicity, safety, and efficacy profiles, and really understanding the immune system in response to SARS-CoV-2 in these models. And this study really just looked at serum samples from two non-human primate species, the rhesus macaque and the crab-eating macaques, to analyze the immune response following infection. And they measured 23 cytokines at different time points post-infection to assess inflammatory responses and disease and follow disease progression. And as you can see, 8 of them were commonly detected. And so being able to measure this, it really allows you to do a lot of cytokine profiling, looking at the immune activation and the potential for a cytokine storm. And so this kind of information makes it really valuable in terms of being able to understand the disease, but also how can the vaccine be effective at mitigating some of these, and dampening some of these responses. Now I'm in the next few minutes, I'm just gonna move over to the clinical side of things. So when preclinical testing is completed, the next critical step is to submit findings to regulatory agencies such as FDA, EMA, and South Africa also have their own regulatory bodies as well, before human trials. And this submission is known as the Investigational New Drug Application, and it is a vital component of the drug development process and allows you to proceed with the first-in-human studies. And the key components that are included in this application are pharmacokinetics and biodistribution, i.e. how is the drug moving in the body? Is it being processed? Any toxicology and safety reports? Are there any adverse events that were associated with this? The immunogenicity profiles that are being elicited by the vaccine, and which is crucial for understanding safety and efficacy. You want this efficacy data that's coming in from your animal models. How effective is it in these models? And how predictive are they for predicting the likely success in humans? But also manufacturing, ensuring that the manufacturing processes meet the GMP compliance standards to ensure that you've got a high quality vaccine and a consistency from batch to batch. And once submitted, the regulatory agencies review the data and if approved, the therapy will proceed to phase 1 of clinical human trials. And if any modifications are needed, the additional studies may be, you will just have to do some additional studies before resubmission. But the ultimate goal of IND clearances is to get approval to move forward with first in humans. And as before, it typically follows a very similar format to biologics as phase 1, phase 2, phase 3. You're looking at the same thing: safety, immunogenicity, and efficacy. And just a brief overview of each of these phases so that you're familiar with it. So phase 1 is the initial stage of human testing for a small group of subjects or the population, usually around about 20 to 100 healthy volunteers that receive different doses of the vaccine to evaluate safety, tolerability, and dosage. And then you're also doing dose escalation studies. What's the dose that a human will likely tolerate? These participants are monitored for any adverse effects. Blood samples are collected and antibodies are measured. In the initial antibody production is measured. Looking at immune activation. Then you move on to phase 2 that you see on the right-hand side. And this really expands to several hundred participants, and you're including a bit more of a diverse population and some with pre-existing conditions to further evaluate safety and immunogenicity. And this phase continues to monitor side effects, any rare adverse reactions, doing a deeper dive analysis of immune responses by measuring antibody levels and T-cell activation. And you're also looking at efficacy here as well, using things like neutralizing assays. But then the step here is you're also starting to optimize the dosage that is likely going to be recommended for first in humans. And you're trying to balance that between what they call the minimum tolerable dose before you get into it, and it's not outside your therapeutic window. And then you move on to phase 3. And this is really kind of the critical stage where efficacy and safety are evaluated on a larger scale. And this involves thousands to tens of thousands of participants from diverse backgrounds globally, ensuring that the results are going to be applicable to a much larger population. And the primary goal is to determine how well the vaccine prevents the target disease. Participants are randomly assigned to receive either vaccines or placebo, and researchers from various institutions globally will compare their data and calculate the vaccine effectiveness to the best of their ability in this controlled environment. And so safety really does remain the most important thing here when it comes to vaccine testing. And then again, once you complete that, it then goes into a regulatory review for the next stage. Now, I'm not going to go into too much detail because it's pretty much the same. In clinical trials for mRNA vaccine development, there's a lot of tools that are used, a lot of methodologies, very similar to preclinical, that rigorously evaluate safety and efficacy and immunogenicity across different phases of the testing of phase 1, 2, and 3. Again, multiplexing technology, serology assays are often used to look at antibody responses, biomarker assays. Neutralizing assays are used to ensure that it's effectively behaving the way it should in humans. PCR, flow cytometry, and again, single-cell RNA-seq is now commonly being used as a profiling tool as well. And then there's also the physical signs, such as temperature, blood pressure, heart rate, things I think you're familiar with, where normally you would see these transient increases in fever, temperature, so forth, a little bit of soreness. How long do they persist? They shouldn't persist longer than a certain amount of time. And these are the things that they're looking for. And so this is just a case study, things about understanding how vaccines behave and how it really applies to in a clinical situation. One of the most important things we've learned about mRNA vaccines is that not everyone responds the same way. Things like prior infection plays a key role in shaping immune response to vaccination. And this is just a study, that was done very early on in 2020 with us, where looked at healthcare workers who had either been previously infected with SARS-CoV or had no prior infection. And what this data tells us is really, it told us a few things. That those who had already been exposed to the virus showed a significantly higher immune response after receiving their vaccine. Individuals with prior infection produced more antibodies post-vaccination, meaning their immune system responded even more robustly. Participants who were seropositive, subjects who already had antibodies as a result of prior infection, the data is not shown here. Just realized I moved forward, sorry. They had a stronger neutralizing antibody level, which is crucial for blocking the virus. And why does this matter? Understanding the patient's infection history can help optimize vaccination strategies, ensuring that the best protection for individuals and communities. So it's very important to have this knowledge as you develop, go through the process of vaccine development. And so what next? What happens? So it's gone through phase 1 to phase 2 and 3. What happens next? So bringing our mRNA therapy or vaccine to the public requires a lot of regulatory approval, rigorous to ensure it's safe and effective. And it involves submitting what is called a biologics license application to these regulatory agencies, and it includes comprehensive data from preclinical clinical trials, also good manufacturing practices, batch-to-batch consistencies, as well as the post-market safety monitoring plans. And this process of once you've submitted, it can take, the review will happen, it'll take up to probably about 10 months before you get an answer. And sometimes in some cases you saw in COVID, it was a lot faster. And once approved, the vaccine is then made for commercial use. Now, once it receives approval, the work doesn't stop there. Phase 4, or post-market surveillance, is critical for monitoring how the vaccine performs in the real world. That's everybody. And while clinical trials do provide that data to a certain degree in terms of safety, efficacy, and large scale, it's in a very controlled, still in a very controlled environment. You can't test everybody. And so this is the goal of phase 4, is you're looking at long-term effectiveness and opportunities for improvements for the vaccine. And so you have several monitoring methods in place. So you've got a pharmacovigilance data that collect reports and adverse events from healthcare providers and patients to detect unexpected trends, long-term studies, especially for chronic disease therapies, following participants for 5 years or more to assess durability and protection and emerging safety concerns. And then any adverse effects that physicians or patients are reporting are shared as well. And this ongoing surveillance allows for updates and refinements for vaccine formulations, ensuring continued safety and effectiveness. And by closely tracking real-world data, you can build confidence in the population that what you're being administered is safe and effective. I mean, it's a process that goes on for quite a long time, and it's continually monitored, and then modifications are usually made. And then I'm just going to finish off with, what are our solutions? How can we help customers like yourself enable vaccine research? So as Merck, we offer a comprehensive portfolio of solutions designed to support every step of the research and development process, from in vitro studies to genomic analysis and protein analysis. And so for in vitro research, we provide tools as 2D and 3D tools, all the consumables that come with it, tools that you can use for this, and biologically relevant models. That's what I'm trying to say. And then obviously, with our partnerships with the Hub and BioVT, these are highly accessible models to help researchers bridge that gap into translational research. And then for genomics, we offer DNA extraction kits for high purity nucleic acid purification, as well as the single-cell RNA sequencing tools. And here we've got the multiplexing tool for better throughput. qPCR, PCR tools, our high fidelity KOD1 PCR master mix and Kappa Biosystems qPCR enzymes. It provides high sensitivity detection for a range of applications. And for those working in using CRISPR tools, we have tools for that as well. And then for protein analysis, we have Western blotting reagents. We've got the MPH Lux casting system that allows a much higher throughput of Westerns. Antibodies for immunohistochemistry. We have a huge portfolio of biomarker platforms and multiplexing platforms, such as multiplexing our assays. And then assays for different rodents, for different rodent species, humans, and complement panels. And then we also have our own service for developing these assays as well. And then all other tools such as ELISpot as well. And so we're really committed to our researchers to empowering them with these tools so that we can help accelerate their research from basic research to preclinical development and to clinical solutions. And with that, I'd like to say thank you.
Thank you so much. Thank you so much, Dr. Tailor. Thank you so much. So we'll hand over to Dr. Toyin now to take, to take over the Q&A session. Dr. Toyin.
Thank you so much, Olutuwan. Thank you so much, Dr. Anitaben. We have a few questions that came in prior to the webinar, and I'll be asking those questions before we have anyone drop questions in the Q&A box. At the moment, we do not have no questions in the box, but we would read out the questions we had received before the webinar. So the first question is, how is the selection? Anitaben, confirm that you can hear me. How is the selection of appropriate animal models crucial in preclinical vaccine research? And what are some limitations of the current model? Did you get the question?
Yeah. Yes. Can you hear me?
Yes, good. We can hear you.
Okay, yeah, I was going to say, I think you're never going to get the perfect human, right, in an animal model. But often what you find, one of the certainly in vaccines, and this is historical, models like non-human primates, they are probably one of the most common models for vaccine research. Why? Because they closely mimic the human physiology and how they respond to vaccines. Mice have been used for a very, very long time, and I think they'll continue to be used for a very, very long time. But the way that they are selected is really how close are they to the human, and how are they likely to be predictive of the response as well. Mice are often used for biodistribution as well as NHPs. Rats are often used for safety. It's a very, it's more to do with the physiological systems within each of these preclinical models that will help assess how predictive it's going to be. Again, that's about as close as you're going to get. Then you can go into the human model as well, because again, you're trying to bridge that gap. You've got your organoid data, which is you're recapitulating that 3D structure. Then you go into your animals, and you go into, and historically, non-human primates are about as close as that you can get to a human. That's why they're used commonly. Well, there was a second part to this question. Or was there not?
The second part. Can
no,
you
I don't think
hear me?
it was. Yeah.
Then what are the limitations? I think you've answered that while you were also speaking.
Yeah. I mean, there's always going to be limitations, right? When you work with animals, you're kind of looking at a very small population, right? And you don't know how a human is going to respond. When you work with mice, the expectation is it's going to behave the same way, right? But most often, these types of models, they have no pre-existing conditions. So you don't know how the world's going, how everybody in the population is going to respond. And that's why it's very important. Those are kind of some of the limitations. We have it in a controlled environment. We know what the physiology is. We know how it's going to behave. But once you move into the real world population, which is why you do the phase 1, phase 2, phase 3, and in that you start incorporating potential pre-existing conditions, so that you can assess this as well. And so it's very important that when we talk about using animal models, we use those models so that you can basically monitor what is an effective dose that is going to be effective and translate it to humans in terms of clinical studies and how it's going to respond. It's all predictive. Everything you do in preclinical is predictive in the hopes that you can stratify and translate into humans. But again, as having come from an in vivo environment, there is a level of control, but in humans, very diverse, lots of underlying. And that's why when you go to phase 4, it's commercially available, but that pharmacovigilance is still going to happen to ensure that they can refine and optimize and make that vaccine better. Hope that makes sense.
Yes, you did. You really made sense, and you really further explained that part, and I'm very sure everyone is pleased with that. We have a question in the chat, and the question says, how many drug candidates are allowed to proceed to the preclinical phases? Did you get the question?
Yeah, so it really depends, right? So what happens, you start off with a huge library, right? And then it will whittle down, depending on when you've assessed the safety, which is one of the biggest things with any therapeutic. You then whittle it down to, you could have 10,000, then you could have, 1,000. And then by the time you get to clinical trials, you're kind of working with about 5, right? So you're whittling it down. Now, with mRNA, in my opinion, I think it's a little bit more different because you kind of specifically designed that mRNA specifically for that disease, right? And so I think it's a little bit easier in terms of getting it through. There probably won't be as many candidates because it's specifically designed for a given disease. Now, when it comes to things like cancer, that's a little bit more heterogeneous, right? Because now you're dealing with a heterogeneous population, what you're targeting. It's a little bit different there. And you don't want to have any off-target effects or anything like it's not doing what it's supposed to do. But with infectious diseases, once you've identified what you're going for, and that's why in COVID you had all these different types of variants. You had these serotypes, the S1, the N. You needed to make sure which one should we be targeting, which part of this protein should we be targeting. So you'll have that. Understanding the disease is important, right? What are those? That's why assays like biomarker assays are useful because they allow you to identify which isotype is going towards which serotype. And from that, you can then develop your mRNA, saying this is what we should be targeting, this is what we should be doing, this is where we're getting the robust response. And then you've got the class switching and all of that going on. That's a session for another day.
Apparently. We have 3 more questions, but we seem to be out of time. So first, I would like to check with Oluton. Do we have time to still ask these 3 questions, or do we want to probably maybe send the questions to Dr. Anitaben, and then she would provide answers and would feedback the team?
Yeah, I can respond to them after.
I didn't get
we
you.
have your permission. She's Dr. Anitaben says she's fine. She can respond to the questions now.
Yes, please.
But we need the approval of yourself and the rest of the team to be sure that we can proceed with the last 3 questions.
Yes,
we have 2 in the chat. You can still ask one. Okay, so we'll take one from a previous question sent before we go back to the chat. Could you share an example of how findings from preclinical studies have directly influenced the design or execution of a clinical trial for a vaccine? Did you get the question, or do you want me to repeat?
Repeat that question. So could I give an example?
Yes, could you share an example of how findings from preclinical studies have directly influenced the design or execution of a clinical trial for a vaccine?
Well, I think COVID-19 was one of them. A lot of that was COVID-19 was on an accelerated path, right? I don't have that data. And the other thing about data is that, some of the data that I showed is on the public domain. So think about the Pfizer one, right? So some of that is on the public domain. And so that was, a lot of that work is done in non-human primates. Now I have to get permission to show some of that data. But what I can tell you is if you think about the COVID-19, that was your classical case in point, right? You had the Pfizer BioNTech vaccine that went through the same phase, that went through the preclinical trials, and work was actually done in non-human primates, I can tell you that. And then that had moved into phase one, phase 2, and 3. So that's your kind of your case in point in terms of moving from preclinical to clinical. And so that really is kind of addressing that question, because I can't really show the data. But that's the process. And it was on an accelerated track. And that's the one thing I think the nice thing about mRNAs, because you can tweak them very, very quickly. I think the timeline for mRNA is going to be shorter compared to biologics, which are a lot more complex and a lot longer, and a lot more costly. But yeah, and that's one of the important things about consensus subjects I'm very passionate about. But it's one of those things where that the regulatory authorities are looking at, who should we be working with? How are we going to move this faster? I think we're going to see some really nice changes coming forward. But yeah, that's a case in point. I think that really paved the way of how vaccines are going to be generated, going from preclinical to clinical.
Okay, so the last question we have, how is the dosage determination process organized, both in phase 1 and phase 2, before the correct dose is arrived at?
Yeah.
Did you get the question?
Yeah, I can see the question here. How is the determination process organized, both in phase 1 and phase 2 before right? So what they will do is it's called the minimum tolerated dose, right? So what they're going to do, and this is where you're looking at toxic effects, you have to assess the therapeutic window. So you're going to be giving continuous doses. You'll have different doses first in different animals. Then they'll start doing what they call escalation studies. So they will continue to give increasing doses as to efficacy here. Is it doing what it's supposed to do? And then as you look at the top, is it still doing what it's supposed to do, but is it now dangerous? So you're looking for a therapeutic window as to it's effective against safety. So one of the critical experiments they have to do very early on, and that happens in phase 1, is finding that therapeutic window by doing dose escalation studies to find the optimal dose before they can go into phase 2. So, and that's a very common thing to do, whether you're working in vaccines or whether you're working in biologics.
Okay. Thank you, Dr. Anitaben. I have a question here that came through another medium. And the question is, what are your thoughts on tuberculosis vaccines for immunodeficiency patients? Did you get the question?
No, yeah. What are my thoughts on, I don't really know about that one. I haven't really thought about it, to be honest with you, but I can think about it and get back to you on that one.
Okay, that'll be fine. Then finally, that'll be fine. I guess you can always, yes.
Yeah,
you can revert to me.
Yeah.
Did you get me? I said you can revert to me, then I will revert to Oluton, and then I guess you can also revert to the person who sends the question.
Yeah. I've not really, to be honest, not really thought about it yet. So I can think about it and tell you what I think. So.
All right, thank you so much, Dr. Anitaben. And then finally, Could you elaborate on some of the key challenges often encountered during the transition from preclinical studies to phase 1 clinical trials? Did you get the question?
Yeah. So, during each phase, and I would say, when they're very strict. The regulatory authorities is getting over that is your big barrier, right? They are the ones that assess. Like we here in the US, we have the FDA, we have reviewers that sit and they will go through that preclinical data and they will be very rigorous and very critical of getting a therapeutic. Should they deem a vaccine predicting poor immunogenicity, poor efficacy, or if there's a safety concern, they will send it back to you. And what they will tell you to do is do X number of experiments. We need you to go to show this, or they will say formulation, right? Reformulation. Where is this coming from? The biggest concern is safety, because you do not want to give any human a vaccine, biological, anything. You do not want an adverse effect. And so that's going to be your biggest challenge. And what will happen with the regulatory is they are very, very strict. They will go through every experiment, every data point that has been collected. And if they deem it's not safe, they will send you back and say, show us this, 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 and this, which will probably be another additional 6 months of work before you can submit again. Some are okay, some are not okay, but I think, if the due diligence really has to be done in the preclinical phase, which is why there's a lot of money being spent in the R&D phase, because you want to make sure before you go into phase 1, you've checked off, you're literally checking off safety, right? If it's efficacious, great. Your therapy can do whatever it wants, but if it's not safe, it's not going through. That's going to be a big challenge. So, but they will read through it, they will critique the data, they will have meetings, they will work with their sponsor and say, these are the things that you are going to have to improve before we can give you an investigational drug license. It's not an easy process. It's very strict about it.
Wow. Well, thank you, Dr. Anitaben. We have some additional questions, but what we are going to do is, because of our time, we will send you the questions, you provide answers to them, and then we'll share with the people concerned.
Yeah.
I believe that should be fine.
About to the, I can't even say it now, TD question. So I'm to think on my head now.
Thank you so much, Dr. Anitaben. Over to you, Olutun.
Thank you so much, Dr. Anitaben. Thank you, Dr. Toyin. It has been an impactful 1 hour, really. The questions kept coming. So we promise to answer them and send it by mail. So as we draw a close to today's webinar, we have a post-test available in the chat box. It has been pinned. And remember, this is one of the webinars in the series. And at the end of the series, you will get a certificate from the African Vaccine Academy and Merck. So this post-test is a we like call it prerequisite, right, to getting the certificate. And we encourage you to take advantage of this opportunity. And to reiterate some of the key takeaways, we've had a comprehensive overview of the crucial role that both preclinical and clinical trials play in ensuring the safety and efficacy of vaccines. So the rigorous process that is involved ensures that everybody is safeguarded, public health is safeguarded, and bringing innovative solutions to combat preventable diseases. I really want to extend our sincere thanks once again to Dr. Anitaben for our expertise, our insights with us today, and also to Dr. Toyin and the entire Merck team and also Bloom Public Health team for making this an outstanding success. Finally, a big thank you to everyone for taking time out of your schedules. We understand today is Thursday, so many things happening during this hour, but you've actually taken time. We have over 100 people on the call, right? So we just want to say thank you so much for joining us, and we'll definitely send you a mail with the link to the webinar, the slides, and also subsequent trainings and webinars as they come up in the series lined up for the year. So thank you so much once again. Thank you for joining us. We are so happy to have you all here, and it has been a wonderful 1 hour, a little bit over an hour. Thank you so much, everyone. Thank you so much. So we'll say our goodbyes here. Dr. Anitaben, do you want to say goodbye? Dr. Toyin?
Take care, guys. Have a wonderful day, remainder of your day. All right.
Yes.
Bye-bye.
Thank you,
Thank you so
to everyone. Bye. See you at the next webinar.
Bye.
Yes, absolutely. Thank you. Bye.
Bye.